to reconnect. I welcome people to the last two talks of the year, and it's my pleasure that to introduce both of you, EUVEN and Carlos. EUVEN is currently a postdoctoral researcher at the Technical University of Munich, well known to us all for its crap internet. Before that, he was a postdoctoral researcher at Justus Liebig University in Gießen, and before that, the Okinawa Institute of Science and Technology Graduate School in Japan. Even if you have been in more institutions, I couldn't find his CV online, and after about 10 minutes of trawling the net for information about you, I gave up. You have also the distinction of being the only EUVEN seminar speaker who has been invited twice, which is a testament to the quality of your work. So without further ado, you have worked on mammalian and reptilian venoms, but also Hymenopteran venoms, and this is the topic that you will talk about today. So thanks for joining us, and the digital podium is yours, EUVEN. You have muted yourself.
But now I have unmuted myself. Do you guys...
You have unmuted yourself, but you are in presenter mode.
Damn it. Hold on. We'll get there.
Now we can see your email.
Nice. That's good. That's always good.
There we go. This looks like your presentation indeed.
That is my presentation, but now you are probably seeing my presenter mode again.
No. This is perfect. We just see your slide.
Okay. Okay.
Cool.
Right. Are we good?
We are indeed.
Nice. All right. I hope that there is no any sound problems, and thanks for the nice introduction, Ron. It's been a pleasure knowing you all of those years while working at all of those institutions. Okay. So today I will present you our results on the bee venom evolution paper, and so basically the Hymenopteran venom evolution, because we kind of had to expand it to check for bees, we had to look for everything. So let's just give some framing. So just some theoretical framework for what we're doing was that, you know, the underlying question is how do new traits and functions evolve? And because we are doing molecular biology, molecular evolutions in our case, and becomes a question of how do new genes come into being. And the usual answer, like, I mean, you probably have heard that multiple times, read it in the papers, is that, especially in the context of venom, duplication has been implicated as like the major mechanism of creating functional new genes, right? So it all goes down to this guy, Suzumu Ono, and his 1970s book, when he basically proposed that genome duplication sparked novel adaptation. And he is the person behind the idea of neofunctionalization, which basically means that right? So you start with a single gene, then you duplicate that gene, and that gene still performs a certain function, right? In our case, this is a Swiss Army knife. So once you duplicate the gene, you will have two Swiss Army knives, and because of the selection regime now is less constrained, because you have two genes performing the same function, each of them has less selection pressure on itself. So each has the same propensity to mutate into something new, in this case, into a wheel. So the process goes like this altogether, right? You start with one gene performing one function, you duplicate it, you have two genes performing ancestral function, selection decreases, and then one gene is allowed to mutate, and then you end up with the two genes with two different functions. Very simple idea, right? So, of course, after 1970s, many years have passed, and so people decided to update that hypothesis. So, for instance, Force proposed the idea of subfunctionalization, where you start with two functions, then you duplicate, and then you basically reduce one gene to one function strictly, and then another function, another gene to another function. And very similar idea of a specialization, where you don't just reduce to the function that you start with, but you even enhance that function, right? But all in all, it's basically the same idea. There's really little difference between neofunctionalization and specialization and subfunctionalization. So don't get confused when you see those words. They all mean the same thing, more or less. But the point is that This idea came into fixation in venom evolution. I mean, this is basically the dogma how it is seen in our field, right? That you start with a physiological gene, and by the way, a small caveat, it kind of implies as if venom is not physiological, which is interesting because venom, of course, is a major part of the physiology of every organism that has venom. But whatever. So we have a gene that is expressed in the body tissue, as if venom gland is not a body tissue. Then you have a gene duplication happening. And then the one of those genes is like, Nick puts here transferred, but essentially what means is that the expression profile of a new copy is changed, and then that copy is allowed to mutate into a toxin, and then it duplicates to increase, its production or generate new forms. So the idea is that we always start with a gene that has some function in the body other than venom gland. Then we create a new duplicant that now is expressed in the venom gland specifically, and then it's restricted to that expression, and that then it mutates into all the toxin proteins further on. But we keep the original one, right? But the question is, do we even need duplication? And the answer is, like, really, do we even need to? And there are, of course, some theories in terms of gene evolution, for instance, moonlighting, where you basically, one gene can perform several functions. Right, and we know that single genes can be co-opted for a new function in venom quite across different animals. So we definitely have that in platypus happening, and in the parasitoid wasps and in Nasonia. So all in all, it seems that we don't really need to really pressure the whole duplication thing further. But the thing is that we really don't know much about the way toxin genes evolve and genes evolve in general, because we have tracked very few toxin genes back to their ancestral groups, back to their body genes. And then even fewer genes we have, deliberately traced them and shown that they actually arise via duplication, not duplication happening after. So those two things haven't been teased apart at all. And then very few have gene phylogenies resolved at the larger taxonomical clade, meaning that we don't really know whether what we see as a duplication is indeed a duplication or is just us mixing orthologs and paralogs, right? So, which brings us to our topic of venom genes, is that the good thing about venom genes is that it's much easier in our field to actually tell apart toxins versus non-toxins. So it's easy to tell apart the changes of function versus, like, for instance, olfactory receptors or some, whatever, Hox genes. Like when they duplicate and evolve, it's very hard to say where the transition of function happens. While in our case, it's almost always we can say, yeah, this is a toxin, this is a non-toxin, and yeah, of course, there is some gray zone, and of course, there are paradigmatic and marginal cases, but in the larger sense, it's much easier than normal. So how do we actually study it? So how do we actually approach that? And the way we approach that, and we should approach that, is by utilizing synteny. What is synteny? Synteny is the order of positions of the genes on the genome. And the reason why it's important is because it allows us to compare orthologs to orthologs or paralogs to paralogs. So say we found that this is the cluster of toxin genes that we are interested in. So what we do first is we trace it back to, the organism that it comes from. In this case, it's viper. And so we trace it back and we find that it's surrounded by the other genes, of course, because it's in the genome, so it exists in the context. So what we do then is that we forget for a second about the genes that we are interested in, and we're looking for their neighbors. And we start looking as wide as we can so that we have the best understanding of the whole evolution of those genes across as many species as we can, right? So again, the idea is that we try to trace it as much as possible, tracing neighboring individual genes. Usually 5 is enough. 10, you would feel comfortable. You can go further, but as you can see, further starts making no sense. Like, yeah, you can find the syntenical pattern, but you essentially are only interested in finding the similar regions for your genes and other genomes, not just figuring out synteny for the entirety of tree of life. By the way, if you do that, tell me, because that will be very good. So In this slide, we did, for example, three-finger toxin study that we recently published. And so what the outcome is basically we get all of this synteny understanding. So we now know that we're talking about the genes that are in the same position in the same genomic region. We can now compare them. And in this case, this is Ly6 and three-finger toxin. So every colored arrow is a gene. Don't try to understand it because we're not here to talk about three-finger toxins. This is just as an example, right? So once you do the synteny analysis, you arrive at the group of genes that then you can further classify into groups using phylogenies and other things. However, there is a certain limit to it. And you still want to have as best a clustering as possible. And that's why we came to AI in both in this study and in bee venom evolution study. So just briefly, the way it works is that there are those things called language models, right? We all hopefully familiar with them at this point because ChatGPT is basically the language model. So what language models do, they understand the context and they understand the structure of information in the non-linear way. So they can understand that the word that comes first in the sentence is related to the word that comes last in the sentence. They don't just check for the sequential position and sequential similarity the way, for instance, if you align sequences, the alignment would just check for the local blocks of alignment, right? But the good thing about language models is that essentially if you find the way to encode any information, you can use them to deal with that information. So in this case, those are even though originally they are made to do with human languages, they were recently retrained to, I mean, recently being like 5-ish years, they were repurposed to work with proteins because essentially you treat each amino acid as a word and a protein itself sequence as a sentence. And the way it works is that you have a transformer model, and I'm not going to go into the details what transformer model is, just for all intents and purposes, magical black box. And so that magical black box has been trained on a large corpus of data. Usually it would be uniprots that is redundancy reduced to say 50 or 60%. Right? So it knows the complexity of the existing or at least known proteins quite well. And it can understand whether how each amino acid, what meaning does it have in the bigger sequence of the protein. So then when you present it with the new sequence, it can essentially do a masked reconstruction. So if you mask a certain amino acid, it will be able to tell you that much likely the amino acids that you masked are this, right? So in this case, the example, it would be sequence spelled with V because we don't have U for amino acid. And so it would try to predict that the next amino acid would be V, right? So you have S, E, Q, and then it would try to predict V. So the way we use it is that for each amino acid within the protein, it would create a vector. And then we can take a mean out of the entire protein length of those vectors, and we will get a vector for each individual protein. And those vectors, they will be essentially like a barcoding. They will be unique, and if the proteins are different, the vectors themselves will be very different. But the closer proteins will have closer vectors. And so we're still talking about the high dimensional space, so we cannot really perceive it. So we have to do dimensionality reduction. This will, of course, mean that we lose some information because we're coming from a vector that has 1024 or higher dimensions and trying to collapse it to something human readable. So we only do that for illustration purposes. If we are, doing any downstream analysis, we don't do that because we will lose some information. So once you will see the graphics further down the line, just bear in mind that this still is a simplification, right? So, but by doing that and by putting a lot of proteins, we can essentially create what people have long wanted to have, which is the sequence function space. I mean, obviously adding fitness, like in this example, is hard because you would need to measure it. And obviously the understanding of a function in this language models, they're still developing. So we're definitely not at the point where we would, want to be, and we will be. But we're already approaching the situation where we can just take all the proteins, put them all in there, have the model process them, have the model understand them and cluster them, and then using that three-finger toxin example, we will get a three-finger toxin sequence function space that will essentially just cluster them based on its model's understanding. And of course, you're at the mercy of the model's understanding and whoever trained the model, but right now they are good enough, so already good enough. So you can see, for instance, that we can already tell cytotoxic three-finger toxins from long-chain neurotoxins from just plesiotypic forms. And what's good here is that not only does it understand features of the molecule and has some inclination of how similar functions relate to each other, but it also is a great tool to show where our straight sequence similarity classification fails, and those here are marked by the olive rhomboids, right? So the olive rhomboid here is non-standard, means that a human specialist, in our case, that was Stephen Aird and Daniel Deshevsky, who are very good specialists, and Tim Jackson, who are very good specialists in three-finger toxins. And they, by looking at the sequence, they couldn't tell what sort of sequence that is. Is that a long-chain three-finger toxin? Is it a short-chain three-finger toxin? Is it something in between? But the model clearly understands. And so the model clearly puts them together. And I will just tell you that we started in a collaboration testing some of them, and we find out that even smaller clusters, they still have some meaning. For example, there is a cluster, one of the smaller short chain three-finger toxin clusters here. We had no idea what it is until we were just tipped from a different researcher at the conference was saying, yeah, they have very different activity, and some of them are not known. So now we can check them for those activity and see that they indeed have that activity. So this is so much better than what we previously were using in terms of clustering proteins and understanding whether your protein, where does it belong to in the larger scheme of things in terms of family, but also just understanding the evolution and family, evolution of gene families. This is like invaluable, because even on this picture, right, and those are on the left you have two-dimensional projections, and the D panel is the three-dimensional projection, obviously again on the two-dimensional space. So even with all of those dimensionality reduction problems, we still can clearly see how the sequence space is being explored after the three-finger toxins lost their membrane anchoring domain and started evolving into all kinds of functions. And you kind of have the least toxic plesiotypic ones in the middle, and then they go exploring other functions in the different directions. So I think that is basically a tool that we should definitely be using more and more. So how does it relate to our bee venoms? So this is basically the gist. And so what did we do? We did a bunch of stuff. So this study is a ginormous study, and I really don't recommend anybody doing it this way because it's just too big. But basically, we started with just scouring all the known information about the bee venoms, and we tried to understand what the bee venoms in general are known, like what the classes and families of proteins are there. Then we looked for all of the UniProt for Hymenoptera. We picked the best genomes of bees and other Hymenopterans as much as we could, which was, clocked up to 32, but out of those 32, 4 I knew, and 1 was just the ones that we had with this publication. We also did some proteomics and transcriptomics, and I tried to do this as precisely as possible. So we were doing not just your normal Trinity transcriptomics assembly, but we were doing multiple assemblers that we were then creating a consensus assembly, and so that we don't miss on individual transcripts. And the proteomics was also highly precise. So with exception of Halictus, those were big bees. So we used Xylocopa violacea, Halictus scabiosa, and Apis mellifera as a control, so to speak. And obviously, Xylocopa violacea is a huge bee, so having venom from it was not a problem. But having venom from Halictus was a bit of an issue, but we still got good results, right? So then we compared it to the genomic information, and we got some stuff out of it. So then let's go in this bit by bit. This is kind of where it started. So it's me in 2019, way before COVID even happened, which was sweet times. And I basically was scouring all the available literature for what is known for each individual bee species in terms of what, and just in general Hymenopterans, what we know of their venom composition. I created this huge table that basically later with the Uniprot data became this figure of the manuscript. And what it allowed us to know is that bee venom is extremely complex thing, and Hymenopteran venoms are very complex. However, there are certain patterns in it. And for instance, you will always see phospholipases. You will always see things that are called venomalogens, and we will have a point in venomalogens in a bit. You will always have venom-serine proteases, peptidases, and so on. Others, not so much. So, meaning we can kind of reliably understand that the majority of the venom should be that. And that's what we call here major achillea venom proteins, which would be all kinds of different small peptides, phospholipases, secapine, venomalogen, venom acid phosphatase, peptidase 4, and serine proteases. Achillea toxins here are in scary quotes because we will touch a bit on them later. So the mass spectrometry basically confirmed that, right? The mass spectrometry just gave us that the majority of those 3 bees venoms, and I mean, obviously Apis mellifera we already knew, that is made out of apomin, melittin, and these major venom proteins, and as well as some small stuff that we really don't know what it does. So we basically decided to zoom on these ones and to zoom on ones that have a clear known function. So we didn't want to go for things that we don't understand what they're doing, like vitellogenin, for instance. Maybe it's just there for contingent reasons, or chitinase, maybe it's there just again randomly. But I mean, obviously, we know what chitinase does, I mean, what it has in the venom, right? So we just concentrated on the known venom components and the ones that are more or less verifiable. So, and then after that, I did a bunch of genomic search. And the way I did that is that I manually looked at each instance of the exon blasts of those venom genes across 32 genomes. I do not recommend doing that. I ended up looking at hundreds of thousands of genes, and I'm not kidding. Because if you're trying to look for serine protease in a genome, you will find all kinds of serine proteases. And in our case, they are the venom serine proteases from the Hymenoptera. They're closely related to trypsins. So you will have all kinds of trypsins, all kinds of hematrypsins, and to really differentiate between them and to really figure out the synteny, you would need to look at them. So I do not recommend. But I did that, so we had things to work with. So once we gathered all the genes via the first blasting and then manual annotating them, then getting the synteny analysis properly, and then having finally the candidate genes, we tossed them all in the protein family to create a protein family tree. And then out of that, we did the embedding space analysis, so that protein sequence function space, to confirm that, right? So just to give you a snapshot, this is the files that I've already processed and already had the candidate genes secured, but because the genomes are broken into scuples, you still need to puzzle them out. So this is just a small snapshot of serine protease, preliminary figure how it looks like. And if you zoom in, you will keep seeing different things. And by the way, they will all be called differently. They will be like Persephone-like serine protease, snake venom-like serine protease, venom protein. Like the names are atrocious. So relying on the names is also not possible. So yeah. All in all, this kind of analysis gave us this sort of figure, which essentially says that which shows that with exception of the peptidic toxins like your apamins and your melitins, and if you're going for ants like your panery toxins, myrmecidial toxins, if you're going for wasps, your mastaperans, vesperkinins, and so on, apart from them, the core machinery, which is out of those venom proteins that we selected based on their prevalence across the entire Hymenoptera, all of them, every single subfamily of them, and I cannot stress it hard enough, like I will repeat it, every single subfamily, not just family of serine proteases, every single subfamily of those serine proteases, venom acid phosphatases, venomalogens, peptidases, they all exist from the beginning of Hymenoptera, which is highly indicative that this is the venom system that hasn't changed in that sense, that the way it started in Symphyta is basically the venom system that the Hymenopterans are still using, which makes it a contender for probably the longest active or most ancient active venom system. Obviously, Nigerians and spiders have something to say about it, but the idea is that these are the same genes, right? Which is crazy stuff. If we look for apaments, for instance, we will get, because the logic goes like this, right? So we figured out where the core venom genes are. We figured out that they are all present everywhere, by subfamilies, and there are some expansion and the contraction of the subfamilies, because probably some of them are using it more, some of it less, but they're all present everywhere. While the small peptidic toxins are not so much. So what happens with them? And what we discovered is that they behave differently, but differently in a way that, for instance, apamins, right? So at the top you have Apis mellifera, and you have your MCDP, mast cell degranulating peptide apamin, and the gene here that is called apamin-like is terterpin. But the interesting thing is that this terterpin is more closely related to every other B venom apamins that we looked in the genomes than to Apis mellifera, MCDP, and apamins. That means that what we think as a defining member of the apamin family is actually in more outlier, that the majority of the apamins, they are more terterpin, they're closely to terterpin. But also, what we previously assumed that most honeybees will have this similar amount of apamins and MCDP, like one gene each, right? That is also not true. There are much more apamin-like genes in the unstudied species, and so the pharmacological potential of that is quite frankly quite insane, because this is just the species that I looked at, and there are more and more and more, right? I mean, some of the genes, we have 32 genomes in the paper. While melittin basically does not experience any sort of duplication. So while apamin keeps expanding and contracting and keeps evolving, and there are new very interesting forms there, with the melittin is just a single copy gene, and it very likely existed before the radiation of the equiliata, but it definitely is more or less one single copy gene everywhere, and they're kind of close-ish to each other. Still slightly different, but very not as much as apamins. So if we go to our machine learning analysis, what we will see is that if we put them all into this protein sequence space, then each family clusters as a separate cluster, with the exception of venomologens, and we will get to them in a bit later. But if we zoom in, we will continue to see the same thing, and that is probably made problematic by the difference between the tree-based classification and what the model understands, because the labels are based on the position in the phylogenetic tree that we put all of the sequences in, while the model obviously doesn't have any inclination of phylogeny, so it just classifies them based on its best understanding and similarity. So it can still recapture most of these families, but there is some sort of a gray zone where maybe either the phylogenetic tree was not certain, or maybe the model is, seeing something that we don't capture with other means. But more or less, we still see the same pattern, and you can clearly see that those multigenic families, they're clustered by subclades, right? Not by species. The species are present in every of those subclades. And yeah, in this situation, the red is an outgroup, so it's okay that it's all over the place, right? So those are acid phosphatases, those are serine proteases, and those are venomologens. Just a quick note on the venomologens. Venomologens are all over the place, and the annotations for them are atrocious. I don't think we even know what venomologens are. Venomologens of all the venom families of hymenopterans, venomologens are the least studied. The genes for them, the annotations range from 2 exons to 7 exons, which clearly indicates that we don't really understand how they should look like, or they're one of the most variable families ever. But also, nobody has ever, figured, like, the way they cultivate venomologens, it's not because they are causing allergy, it's because some of them are allergenic, in humans, and but every single protein in the honeybee venom is allergenic in humans. It's just others have other names, but venomologens didn't get other names. So they're still called venomologen 3, venomologen 5. even though if you see those names, they mean absolutely nothing. Venom allergen 3 is the same as venom allergen 5, so don't even bother putting 3 or 5s there, okay? Venom allergens are a mess, so we need to figure them out more. So the kind of getting closer to the final point, and I see my time, that's okay. So the obviously the elephant is in the room is the E. coli toxins. So I mean, I'm sure some of you are familiar with, or hopefully most of you are familiar with Sam Robinson's paper on the Myrmecia gulosa, which is a great paper. It's really nice, like well written, nice study. The problem though is that they proposed there by this figure alone, and I'm not kidding, they don't provide any other justification. They just say, here is the figure, and on that figure you can clearly see that the signal peptide and propeptide of the ant peptides, which are poniritoxins, pilosylins, myrmecetotoxins, wasp peptides, which are vespakinins and mastoperans, and honeybee, or rather bee peptides, which are your melitins, that they all share signal peptide and propeptide, even though the mature peptide doesn't look like anything alike. I gotta say, from my personal perspective, the propeptide and signal peptide also do not look like similar, but we can take that as an argument, right? The issue, of course, is that this is the only evidence for the proposed hypothesis, and then everybody runs away with that hypothesis and say, yeah, this is proven and shown to be the case, which it isn't, because it's still just a figure in the paper. So what we did is we put all of those proposed E. coli toxins, and we did that in the various data sets so that we know that what we're seeing is correct. And basically what you see is that they don't form a cluster. They form a subclusters by species, which clearly indicates that they're not really a family in that sense. And they also would invade into other families based on how you define the E. coli toxins. So, but if you try to investigate the claim that it's the signal peptide that makes them more similar, then the machine learning shows the reverse is true. That it shows that if you leave the signal peptide intact and just look at the molecules with the signal peptide and the mature peptide and propeptide, of course, then you will see clusters by clades, right? Which just clearly separates clades from like ants from bees from wasps. However, if you remove the signal peptide and just have everything else, then you start seeing the admixture, even though it's still separated by clades, but it's not as dramatic. And so this is, one line of evidence. And then if we look at the positions of the genes, we will see an additional line of evidence that also shows that we are quite likely that E. coli toxins do not exist in the way they are proposed. Which is that if you look for a melittin region in the wasp, you will find nothing. But if you look in the ant, you can find a gene that is pseudogenized, and it's kind of looking like a melittin in ant. And for mastoperans, you can actually find pseudogenes for mastoperans and ants, for mastoperans and bees and ants in the respective parts of the genomes. And poniritoxins is kind of similar in that situation. So
time is almost up.
Yeah. I'm like one slide before the thank you slide, so we're there. So the point is that even it's possible that they form a family, but they definitely don't form a monophyletic family at the level of Aculeata. They might form a monophyletic family at the level of Hymenoptera. They might be related, don't get me wrong, it's hard to say otherwise. But what is clear is that at the level of Aculeata, they already represent 3 distinct groups of proteins, right? So calling them E. coli toxins becomes slightly weird. We can call them Hymenoptera toxins, but they definitely are not the same gene at the stem of Aculeata radiation. Now, just to wrap it up, basically what we see is that the Hymenoptera venom arsenal is insanely old, and that it exists starting from a symphyta. And so basically the same machinery that symphyton wasps use to induce a gall and create the biochemical physiological change in the tree for the larvae is the same machinery that then parasitoid wasps use for to inject their larvae into others' larvae, and it's still the same machinery that modern aculeates use to sting us and to cause pain. What changes is the peptidic component, which is kind of the major component of the venom, but everything else is kind of delivery system. So we can compare it to ballistic missile, where missile itself stays the same. We just change the warheads depending on kind of a species and conditions that we're in. So, just the conclusions is that venom genes of predate Aculeata as a clade. Aculeatoxins are likely to be polyphyletic, and there are still hundreds and hundreds of unknown toxins that we can get from the genomes that will have us very happy in terms of their pharmacokinetics and such. So yeah, would not be possible without all of these people, and of course, without you listening to it. Thank you.